Okay. I'm gonna show you the first room. The introduction. It's right over here. Here we are.
Throughout history, there was an argument between Lamarckian evolution theory and Darwinian evolution theory. Lamarckian evolution theory states that changes arise through use and disuse. This supports the post-exposure hypothesis. Darwinian evolution states that random inherited mutations and natural selection are the driving factors for evolution. This supports the pre-exposure hypothesis. Delbruck and Salvador Luria were two scientists looking to solve the debate between the evolution theories. After meeting at a conference of the American Physical Society at Philadelphia, they decided to work together. The idea for their experiment arose from Salvador Luria watching a slot machine, and he noticed that the slot machine doesn't behave statistically, and applied this reasoning to bacterial mutations. The experiment took place in the 1940s using E. coli bacteria and bacteriophages. They expected the mean and variance of the colonies to be similar. They concluded that mutations were random and not directed by the mutation, favoring the pre-exposure hypothesis, and thus Darwinian evolution. This experiment marks the birth of bacterial genetics. Rather than using E. coli in our experiment, we used yeast cells, and rather than using bacteriophages, we used cannabinoid. The pre-exposure hypothesis relies on mutations arising randomly before being exposed to a pressure. In this hypothesis, there would be a high frequency of red colonies, creating jackpot plates. The colonies on these plates should all have the same mutation, with different mutations between plates. The post-exposure hypothesis relies on the mutation arising after exposed to a pressure. In this hypothesis, there would be a low frequency of red colonies, and each colony would have a different mutation. The parental yeast strain used in the experiment contained 3 existing mutations. CAN1 encodes a plasma membrane arginine permease. A mutation of CAN1 renders the cell immune to cannabinoid. An AD2 mutation causes red pigment to accumulate in the cells. Sub4O encodes a mutation suppressor of tRNA. If Sub4O has a mutation, then both mutations of CAN1 and AD2 can exist, and the cell is immune to cannabinoid and red. If there's no mutation in Sub4O, then the mutations of AD2 and CAN1 are suppressed, and the cell is no longer immune to cannabinoid.
Let's go to the next room. Here we are. The methods room.
Moving on to methods, we had 2 different jack plates. One was labeled A and one was labeled B, and then we had 8 different C plates. From each of the jack plates, we took 8 different colonies from each and put them into PCR tubes so that it had A1 through 8, B1 through 8, and then C1 through 8, all in different PCR tubes for a total of 24 total cultures. We took these and lysed them to extract the DNA from the yeast cells. Then we centrifuged this to form the supernatant. This supernatant was then pipetted into clean PCR tubes, and then a master mix containing primers specific to the SEP gene were placed into the PCR tubes with the DNA, and then a PCR was conducted on them to amplify the DNA. We then took this PCR product and we needed to purify it with a series of buffers, which are listed on these slides in more detail for further use. The PCR products that were purified were then put into a gel, and gel electrophoresis was conducted to ascertain if there was in fact DNA present from the amplification. This was then confirmed it so that we sent the DNA off for sequencing at our collaborating DNA lab for results.
Okay, we're going to the results. Let's go. Here we are. Come on inside.
Figure 4 depicts electrophoresis of purified DNA. DNA is present in this assay for A1 through A8 at 750 base pairs. DNA is also present for C3 and C4 at 750 base pairs. No DNA is present for B1 through B8, as well as C1, C2, and C5 through C8. Figure 5 illustrates DNA sequences with the accompanying mutations in comparison to the normal SUP4O sequence. Highlighted letters represent where a mutation is seen, except in A7 and A8. In A7 and A8, no mutation is detected because base pairs match with the specific SUP4O sequence. In the rest of the jackpot A samples, a mutation is seen replacing thymine with adenine. At a different mutation point, C3, guanine, is converted into adenine. In C4, adenine is replaced with guanine. The dashes represent identical base pairs as in SUP4O.
Last but not least, let's go to the discussion. Here we are!
The purpose of our experiment was to analyze the DNA sequence of specifically the SUP4O gene to support the pre- or post-exposure hypothesis. To do this, sequences were examined for all the same or different mutations for colonies on a variety of plates. For the pre-exposure hypothesis, as discussed earlier, the mutation would occur to cause greater number of red colonies, and if they were on the same plate, would have the mutation of the SUP4O gene at the same location. For the post-exposure hypothesis, there would be less red colonies, and the colonies on the jackpot plate would have mutations at different locations. On A1 through A6 plates, the mutation occurred at the same locations, showing that they are identical from descent, like the pre-exposure hypothesis. The A7 and A8 also were taken from the jackpot plate A, but did not have the mutation of the SUP4O gene at the same location, but did have the mutation of the CAN1 gene to survive on the canavanine medium plate. The C3 and C4 plates were both from separate plates, but were red colonies. The genetic sequencing showed a mutation of the SUP4O gene, but at different locations. This shows our sequencing results all support the pre-exposure hypothesis. To improve this experiment, we could add a third jackpot plate. That way, if we were to have an additional experiment where one of the samples are unable to obtain DNA, we have an additional jackpot to gather DNA from to support our results. Further research can be conducted to look at why the A7 and A8 plates are able to be read without having a SUP4O mutation.